So great to be at an in-person meeting. Some people here. So I think you probably all get this question when you work in aging research. People ask you, what are you doing to live longer? And you might be surprised that when people ask me that question, I don't usually say the ketogenic diet. I usually say exercise. I'm an avid triathlete, ex-international rower. And that's actually how I came to study ketone biology. So I was interested in how we could use exogenous ketones to mitigate the extreme stress that occurs during endurance exercise. And sort of through my career, that interest has transitioned into how some of those same stresses that occur during aging could also be modulated by exogenous ketones specifically. So as you are all aware, I'm sure, and you've heard some of the other speakers say, caloric restriction is a very, very robust way to extend health span and lifespan across multiple organisms. And this occurs through hitting on many of those hallmarks of aging, which again, I'm sure you're all familiar with. But really one of the key metabolic switches that happens, especially in higher order organisms, is the change from glucose metabolism to fat metabolism and also ketone production as well. And so interestingly enough, at the same time, in the same issue of cell metabolism, two independent research groups, one at UC Davis and one from my mentors at the Buck Institute, John Newman and Eric Verdin, both showed that putting mice on a ketogenic diet could extend lifespan, but also importantly extend health span, so physical and cognitive performance in these aging animals as well. And so what we've been focusing on since they made this discovery is really the biological mechanisms whereby ketones themselves may be extending lifespan and health span. So just to make sure we've all gone back to basics, we'll just cover here the three biological ketone bodies that occur, because some people who study chemistry, ketones are more of a general term, but in human physiology, ketones refer to acetone, which is the one we can smell in the breath of people who are in ketoacidosis, acetoacetate, and beta-hydroxybutyrate. And these are the two that circulate in our blood, BHB. And really BHB is the easiest to measure, so a lot of work is focused on that. And I'm personally really interested to discover more functions of acetoacetate as we get better at measuring it. So traditionally, ketones have been thought of as an energy source. So when we're in this state of carbohydrate restriction or caloric restriction, our bodies will make ketones, fat, as an alternative energy to glucose. But really, in the last, I guess it's getting on for 10 years now, we've discovered that the molecule BHB can actually bind directly with proteins, behave a little bit more like a drug, and have signaling functions that affect multiple cellular processes, and many of those intersect with aging biology. So how do we get ketones in the blood? So firstly, it's kind of what we've just been discussing, the process of endogenous ketogenesis. That occurs in conditions of low carbohydrate availability, whether that's in conjunction with caloric restriction or in sort of isocaloric state, but a ketogenic diet when dietary carbohydrate. In mice, it has to be virtually zero, but in humans, it can be around about 50 grams of carbohydrates. It's traditionally kind of the accepted threshold for getting a human into ketosis. So in that case, blood insulin falls, lipolysis increases, and those free fatty acids are used as a substrate in the liver for ketogenesis. One little nugget I want to throw in is that recently we've actually discovered that other tissues are locally producing small amounts of ketones in the brain and in the heart, tiny amounts, and in the gut as well. And this may be having an interesting function in modulating aging signals in those tissues locally. But when we think about ketogenesis, we mainly think about the liver. So if you're not going to make your own ketones, why don't you drink them? And this is actually what I've spent most of my career working on, developing these different exogenous sources of ketones that you can consume. And within hours, they will put you in a state of hyperketonemia or elevated ketones in the blood without any other changes in the diet. Now, this is only transient. So you'll take one of these, your ketone levels go up, and it's a little bit like a blood glucose spike after a meal. It'll go up, and then a few hours later, it'll come down. These are the three main types of exogenous ketones that are in use at the moment in consumer products in the lab. Ketone esters, which consist of ketone precursors joined by an ester bond. And we eat esters all of the time. So our guts can very easily break those ester bonds and release those precursors into the blood. Ketone salts consist of ketones joined by an ionic bond to a mineral, and ketogenic agents you can see on the far right there, medium chain triglycerides and the ketogenic alcohol 1,3-butanediol. I like to call it the party ketone. It requires very little metabolism to convert these two things into ketone bodies. So they're not directly ketones themselves, but very readily converted. Now, I don't have time to cover it today, but there's an awful lot of differences between these different types of compounds in terms of efficacy, ease of manufacture, all of those things. But I'm happy to discuss that with anyone over the course of this meeting. But that's just an important point to bear in mind when thinking about using these tools. So once you get ketones into your bloodstream, it doesn't really matter so much where they've come from because they're going to be behaving in a very largely similar way. So BHB, you can see there in the blood, is taken up into many extrahepatic tissues through monocarboxylate transporters, which ubiquitously expressed. And there's a very simple metabolic process to convert ketones to acetyl-CoA, and they're using the Krebs cycle to fuel ATP generation. And so this is the first place where ketone biology intersects with aging, because mitochondrial dysfunction is one of the key hallmarks of aging, and we know that the mitochondria get less good at producing fuel, and some tissues go into energy deficit because of mitochondrial dysfunction. And actually ketones in some cases can bypass that, the dysfunctional steps. So we have ketones use as a fuel here, but also ketones use as a signal. Now this is really busy diagram, so if you're interested in this, I'd encourage you to go and read the 2017 review that was authored by my two mentors at Buck, John Newman and Eric Verdin. They go into all of this in a lot of detail, but I particularly want to call out the three that are assigned here with the hallmarks of aging, because we know that ketones can affect intracellular communication through modulation of inflammation. We know that ketones can affect epigenetics, and we also know that ketones, recent discovery was that ketones may interact with senescence pathways as well, and we'll talk a little more about that later. But before I move on to give you some examples, I want to re-emphasize again that the ketogenic diet and the state of ketosis that comes when you consume exogenous ketones are very distinct and different. The ketogenic diet, you have so many other things going on as well. You have the low insulin state, a higher fat intake in some cases, lower carbohydrate intake. And so when we're looking at ketones as an intervention, we have to consider these two things as quite distinct. But the thing that is common between the two is the elevated ketones in the blood. And so really, rather than focusing on carbohydrate restriction per se, a lot of what I think about is direct effects of ketone bodies as a fuel and as a signal. So first up, we'll talk a little bit more about how ketones can be used as geroscience metabolites to affect tissues where mitochondrial function occurs, because there's some good clinical examples here, an area of quite active research and advanced research as well. The first tissue where we know that ketones can help rescue metabolic dysfunction is in the brain. So on the left, you can see glucose uptake in the brains of control individuals, individuals with mild cognitive impairment, and then individuals with Alzheimer's disease. You can see it going from yellow down to green, and that indicates decreasing glucose uptake in these people's brains. Whereas on the right, you can see ketone uptake, acetoacetate, so it's lower in the controls, actually increases through with people with Alzheimer's disease. This work was done by Stephen Cunane's group up at the University in Sherbrooke, Canada. And he went one step further than this and supplemented for 6 months with exogenous ketones. And his group was able to see that ketone supplementation was able to rescue low brain energy status in individuals with cognitive impairment. And they also saw some improvements in cognitive function that interestingly correlated directly with the levels of ketosis that each individual was able to attain. There's a whole body of animal work showing, recapitulating these results, which I don't have time to go into here, but also suggests that ketones may have other functions in neurodegeneration. And I think this is an area of a lot of promise. It's just a shame that the Alzheimer's drug field is full of, really just a graveyard. So I think it's going to be difficult to move this forward, even though the data is very promising. The second area, the second tissue where metabolic function is compromised, especially with age and disease, is in the heart. Age is a big risk factor for the development of heart failure. A while back, we made the observation that ketone body metabolism is increased in the failing heart, and the heart is a metabolic omnivore, it really eat any substrate that's available, but the failing heart starts to bias itself towards ketone metabolism, and a series of elegant knockout experiments showed that this was in fact adaptive. Recently, this work has been extended to show that ketone ester supplementation in both rats and mice and different ketone esters as well. So this is a replicable effect. These were able to not only prevent the development of heart failure in certain animal models, but also rescue some phenotypes as well. So from that promising animal research, people have started taking this into clinical studies, and the data you can see on the right there is from an acute infusion of ketone salts into individuals with heart failure. You're looking on the sort of left there, that's cardiac output, stroke volume all going up acutely, and the red box to the right there is stroke volume and other measures of peripheral resistance going down with ketone infusion. So obviously this is somewhat limited because it's an acute, very acute setting, and there's some work to be done before we can translate that out long term, but it's certainly encouraging that we're seeing findings translate from animal models up through to humans as well. So I'll finish by talking a little bit about these signaling protein binding effects of ketones, focusing on epigenetics, intracellular communication, and cell senescence, as I mentioned earlier. So one of the first protein binding effects of ketones that were identified was actually found by Eric Verdin, as I said, my mentor at the Buck Institute, and John Newman as well. And they found that BHB was an endogenous histone deacetylase inhibitor, interacting directly with HDACs, increasing acetylation and increasing the expression of genes involved with resistance to oxidative stress, which obviously has multiple implications for aging process. A more recently discovered epigenetic alteration is the attachment of BHB itself to histones, the binding of BHB to histones, which is called BHB elation or histone beta-hydroxybutyrylation, depending on who you speak to. And this is a lot less well characterized and has not been replicating quite so many settings yet as the histone deacetylase activity. But when it is seen, it's seen in association with an increased expression of fasting related genes. So I expect we'll see more BHB elation research coming out in the near future. Intracellular communication. So Deep Dixit, who's here at this meeting later, his group was among the first to show that BHB can interact directly with a component of our innate immune system, which is called the NLRP3 inflammasome. I always think it's a really beautiful part of the immune system there. It's activated on response to damage signals, and when it's activated, it sets off a cascade of inflammation, release of cytokines, and this is implicated in multiple diseases of aging. And BHB interacts directly with the NLRP3 inflammasome to prevent its activation. And so in multiple preclinical models, this effect has been replicated and shown to have an inflammatory effect. And so possible implications here for ketones decreasing inflammation. And finally, another recently discovered direct protein interaction of BHB is its binding to a protein called HNRPA1. This protein stabilizes the mRNA for OCT4, which shifts the balance of cells in the vascular system away from senescence and towards quiescence. And you can see on the right there imaging that stains for markers of senescence, and you can see that they decrease with increasing concentrations of BHB. So this is again novel, interesting, and there's another group that's done similar work looking at stem cell function in the ducts, and seeing similar results with ketones as well. So again, earlier stage than all of the histones and energy functions that I mentioned earlier, but I think for me as a researcher in this field, it's exciting to see the binding partners of BHB being expanded. So sort of a summary of the concepts, we discussed that ketones are both a fuel and a signal, and that has multiple implications for supplying fuel to the aging brain, aging heart, as well as signaling across multiple tissues as well. I would like to call out that we desperately need more clinical studies in this area. A lot of my work with exogenous ketones is hoping to facilitate that, because we have now compounds that we can safely administer to people of different ages across a range of doses. But we really need to nail down some super critical questions like, where is the threshold for circulating BHB for these effects? Do we have to get people up very high? Or there's interesting circumstantial evidence with SGLT2 inhibitors, which produce very low level of ketosis, and they're actually seeing cardiovascular benefits there, and we wonder if ketones are implicated in that. So maybe the threshold is not as high as we think. But I'm excited to be helping to bring these tools out and part of developing these more targeted, optimized interventions in the future. So I'm looking forward to taking questions on the panel. You can find me on Twitter. I'm always happy to answer questions, and I would like to acknowledge John Newman and Aaron Verdon, who are my mentors at the Buck, and have been fantastic with me there the last couple of years. Thank you.